Hey everyone, my name's Gabe, and today I'm diving into two fascinating pillars of modern biotechnology. Molecular cloning and transgenic technology. So hopefully by the end of the video, you'll know how scientists copy DNA and create organisms with new traits. So first, molecular cloning. When I say cloning, it's just most people imagine copying an entire animal, but molecular cloning is much more precise, and I'll get into that now. So it's about isolating a gene of interest and making multiple identical copies of that gene. So in simple terms, I take a piece of DNA, so for example, from bacteria, a plant, animal, or even viruses, and insert it into a circular DNA molecule called a vector, often a plasmid. And then this plasmid acts like a delivery vehicle. And once I insert the gene into it, I transfer it into a host cell. So usually like E. coli or something like that. And then these cells will grow, divide, and naturally copy that plasmid DNA, giving me millions of copies of my gene. So it's basically like photocopying DNA, but using living cells instead of a machine. So this is how it works. Step one is you select your gene of interest. Step two, cut both the DNA and plasmid with restriction enzymes. Step three, paste the gene into plasmid using DNA ligase. And then step four, insert the recombinant into the host cell. And step five, grow the cells in order to amplify the DNA. So now, let's get into transgenic technology. Instead of just copying a gene, here I actually insert a foreign gene into an organism's genome so that it's expressed as a new trait. So this is how we get it. So like BT cotton and BT corn that resist pests, or glowfish that glow under UV light, golden rice enriched with vitamin A, or transgenic mice using cancer and immune research. So the inserted gene is often called a transgene. Scientists use viral vectors, gene guns, microinjection, or the CRISPR-Cas systems to integrate it right into the host DNA. Once inserted, the organism starts producing the protein coded by that gene, just like its own. Why it matters? So these technologies have changed medicine, agriculture, and research pretty much forever. We can produce human insulin using cloned genes and bacteria. We can boost crop yield. We can reduce pesticide use and even create animal models to study diseases like Alzheimer's and diabetes. So that's pretty much all I have. That's pretty much molecular cloning and transgenic technology summed up. It's just copying genes and engineering life with precision. So, thanks for watching, and I hope you learned something new.